All right, so welcome back to the urinary system. So we're going to finish up this section by taking a look at tubular reabsorption and secretion, so what's going on in those nephrons, and how they play a role in forming dilute versus concentrated urine. All right, so why do we actually care about concentrating our urine, right? Why not just produce lots of dilute urine? Well, we need to conserve water, right? We basically filter something like 180 liters of water a day. Our kidneys do. I mean, 180 liters. I mean, think of your liter bottles and like 180 of those. But we only, pee about 1 to 2 liters a day, right? I mean, give or take, maybe a little bit more if you're drinking particularly a lot. But I mean, can you think of that? I mean, can you imagine that if we weren't able to concentrate our urine, we would basically be tied to a water source. We wouldn't be able to be on dry land. Okay, so it is very important that we're able to concentrate our urine while at the same time being able to filter our blood. So let me just give you some dehydration rates as well, so why it's also important to concentrate our urine. So on a 45 degrees Celsius day, so that's about 113 degrees Fahrenheit, which we're cruising into slowly but surely here in Arizona. So if you're just walking around outside, without rehydrating yourself, in about an hour, you've lost something like 2% of your body mass due to sweating and evaporation. So about at that point, you're kind of starting to feel thirsty. After about an hour out in that kind of temperature, but for the most part you're kind of okay. However, by 2 hours, you've lost something like 4% of your body mass, and at this point you're really tired, you're cranky, you're done with the hike, you're moody, you do not want to go on. Usually at this 2-hour mark is where people who are not familiar with Arizona heat and are kind of dry climate, this is where they get themselves in trouble. They go out on a hike, they go out 2 hours without enough water, and at this point they're kind of tired, cranky, they turn around, and now, if you're 2 hours out, you're 2 hours back, and at about 4 hours, you've lost about 8% of your body mass, and basically at this point you're kind of confused, like you're not thinking straight anymore. Your tongue is starting to swell in your mouth. You got that cotton mouth kind of feeling. This is why a lot of times people that turn around on a very well-marked path to go back to the parking lot end up veering off the path, because they get kind of confused. They start seeing things. And then about 5 hours, you've lost about 10% of your body mass. At this point, you're really having a hard time taking care of yourself even. You're probably dragging yourself on the ground at this point. Your muscle contractions are going to be very clumsy. At about 6 hours, you've lost something like 12% of your body mass. You're basically, if they, at this point, they have to give you IV fluids. You couldn't even swallow if you wanted to, with 12% body mass water loss. You can't take care of yourself at all. So if something came up and tried to eat you, it would eat you. And at about 7 to 8 hours, at 15% body mass loss, you're basically dead. Your body can no longer regulate its own temperature, and basically, breaks down all sorts of proteins in the body. Neurons no longer firing properly. Heart does not work properly, and basically you die. So that's, fairly quickly we can dehydrate, right? So again, it's very important that we try to conserve as much water as we possibly can. So again, those kidneys are going to work really hard at trying to make sure that we're only losing the amount of water that we absolutely have to. All right, so how does a kidney concentrate urine? Well, it's via this osmotic gradient in that renal medulla. So if we take a look at this image here, here's a cortex in this lighter color, and then there's a medulla in that purple color. So if you notice, in the cortex rather, the osmotic gradient, the osmotic pressure is about 300 milliosmoles. Milliosmoles is the unit of measure. So basically that's sort of like a solute concentration in that area. So it's at 300 milliosmoles, and then as you go further into the medulla, it gets higher and higher. Okay? And what we're going to see is, because the solute concentration out here, out in the actual kidney, the actual tissue, is fairly high, that's going to exert like concentration gradient, right? If you remember what a hypertonic solution would do, it would kind of draw the water out of a cell. Well, what this is going to do is it's going to draw the water out of those kidney tubules. All right, so let's just take a look at this. So here we are again, we've got our afferent arteriole, the glomerulus, and water is going to basically flow out of the glomerulus or filter out the glomerulus. It'll be caught by that Bowman's capsule, travel down the proximal convoluted tubule, and then as it goes down the descending limb of the loop of Henle, and I don't know how well you see that actually because that blue is kind of blocking it, but you have aquaporin channels. So you have very specific, those aquaporin channels, so those water channels are inserted into the membrane of cells in this descending limb of the loop of Henle, which allows water to be pulled out of the tubes. Again, just like a hypertonic solution would pull water out of a cell, the hypertonic space in the medulla is going to pull water out of that loop of Henle, right? So what you see here is that in the tubules, right, in this cortical area, it's about 300 milliosmoles, and then as we go down, and here's the numbers here on the left-hand side, the concentration is going to mirror pretty closely it's going to get more and more concentrated. It's going to mirror the concentration found out here, right? And then what ends up happening on the ascending limb of the loop of Henle, now we have no aquaporin channels. Excuse me, so what that means is that water cannot leave this ascending limb of the loop of Henle. However, we have symporters, and if you remember back, symporters mean that more than one ion or molecule are going to get transported in the same direction. And in this case, what you have is a sodium potassium chloride symporter. So this is going to basically actively remove sodium, potassium, and chloride out of the ascending limb of the loop of Henle. So what you see here is that the concentration inside the tubule is going to get less and less, right? Because we're removing the solutes. We're leaving the water there, but now we're going to remove the solutes, right? So basically that decreases the osmotic gradient inside the tubule, okay? These solutes then go on to help create the osmotic gradient here in the medulla. Now by the time we get to distal convoluted tubule, we can add in aquaporin channels, so water can leave once again, so we reabsorb some of that. And then here in the collecting duct, once again, we can add in aquaporin channels. So as that collecting duct goes through that concentration gradient in the medulla, once again, water is going to leave. It's going to be drawn out by that hyperosmotic, in this case. It's not technically hypertonic, it's not a solution, but that hyperosmotic conditions in the medulla. Okay, so hopefully that makes sense to you. So water leaves in the descending limb, right, making the filtrate in the loop of Henle more and more concentrated. And then in the ascending limb, we're losing our solutes, which makes it more and more dilute. But then in the collecting duct, it becomes concentrated once more. Okay, and again, particularly here, the water doesn't just kind of build up here in that interstitial space of the kidney. It will be picked up by those blood vessels. All right. So when we filter things out of the glomerulus, some things get filtered out, I should say, into that Bowman's capsule that we didn't necessarily want to have filtered out. So we have to reabsorb those. And really, most of the reabsorption is going to occur at that proximal convoluted tubule. Okay, so the ways that we reabsorb things, you can either have it paracellularly, meaning it goes between the cells. So for example, there's some sodium that will go between the cells and right into the capillaries. Or there's what's known as transcellular, so it simply moves through the actual cell into the peritubular capillary. So again, it's the proximal convoluted tubule is responsible for most of the reabsorption. So you see about 65% of the potassium, sodium, and water. Okay, and then sodium can either diffuse through, as we saw, it's imported with glucose into the cell and then transported out of the cell into that interstitial space via the sodium-potassium pump. So that's sort of what you saw here. We're just missing that glucose um, symporter as well. And then water is simply going to leave the proximal convoluted tubule via osmosis. Okay, again, as long as we have the aquaporin channels in place. Small ions will leave via simple diffusion. And then your glucose as well as amino acids will be taken back up by symporters. Now, it says here, unless there's too much of either in the blood, as maybe in the case of diabetes. So, for example, we're kind of back to that Lucille Ball chocolate factory kind of situation where glucose has to have this symporter. It has to have this transporter that's going to take it from that filtrate back into the bloodstream. So if there's too much glucose, those symporters can be overwhelmed, and then you won't be able to retrieve it from the filtrate. So basically you're going to end up losing some glucose in your urine. Now, stories have it that one of the oldest methods to tell if someone had diabetes was to actually taste the urine, and if it tasted sweet, you knew they had diabetes. Thank goodness for modern medicine. Okay, that would have been a terrible job. Also, vitamins can be reabsorbed at the proximal convoluted tubule. Okay, so I already mentioned this. So glucosuria, basically those symporters cannot reabsorb that glucose fast enough. And usually if glucose levels are above 200 milligrams per milliliter, some of it ends up in the urine. We have these test strips these days that can measure for that kind of thing. I mean, incidentally, you could, I suppose, do this yourselves. Don't ever. But if you ate like a bag of candy corn or something really fast in one sitting, it would probably overwhelm your kidney transporters at that point too. It'd probably also give you like a super sugar high, so don't do that. But you could probably force that to happen if you really wanted to. All right. So again, in the descending limb of the loop of Henle, the only thing that's going to leave is water, right? In the ascending limb of the loop of Henle, we have this chloride, potassium, sodium symporter that's going to help move those things into the interstitial space. In the early distal convoluted tubule, the cells are simple cuboidal. It will transport sodium into the cell, and then it'll transport sodium via these leakage channels, so that simply meaning that these channels allow sodium to move from where it's highly concentrated to less concentrated, and then the sodium-potassium pump will move it into the interstitial space. There is some more water that gets reabsorbed in the early distal convoluted tubule. Calcium is also reabsorbed, and that'll be directed by parathyroid hormone levels, and then this is the area where certain drugs will be secreted or added into the filtrate, into what will become the urine. So penicillin, for example, and some other drugs that we commonly drug test for. In the late distal convoluted tubule, along with the collecting duct, we have two different types of cells. We have principal cells, and these principal cells reabsorb sodium and secrete potassium. And then you have intercalated cells that reabsorb potassium and bicarbonate and secrete hydrogen ions. So this is one of the ways that we try to help regulate sodium levels, potassium levels, as again here's potassium levels, and hydrogen levels. Okay, this helps regulate pH. So for example, if your pH is too low, you are going to want to reabsorb more bicarbonate to help buffer that, and you're going to want to end up secreting a lot more of this hydrogen ions. Okay. So by the end of that distal convoluted tubule, about 95% of all the solids, water, everything we need to be reabsorbed has already been reabsorbed. And then finally, it's in the collecting duct that we make those final adjustments. So this is just a summary of everything that we have talked about in those slides. If it scares you, don't look at it, okay? But this is just, again, it's just a simple summary slide. So in the proximal convoluted tubule, we reabsorb about 65% of the water. In the loop of Henle, about 15. The distal convoluted tubule, about 10 to 15, and then the collecting duct from 5 to 10 with within the presence of antidiuretic hormone. And we're going to talk more about antidiuretic hormone in the next lecture in the pH ions, so I'll hold off until then. All right, that's about it. Again, so if you have any questions, please let me know. See you soon.